and Erkki is going to be talking on guidance systems for nanomedicines. Erkki?
Thank you very much. It's an honor to have been asked to tell you a little bit about my work. I work on guidance systems, as it says there, for nanoparticles, which then are contain drugs or diagnostic agents. And when you think of guiding a nanoparticle, say to a site of a disease, the first thing you have to accomplish is to get it to the vasculature of that disease site, because the vasculature is available for the blood, and it's the gateway to the tissue. So we probe the vasculature using our a method that we developed 20 years ago. And it goes like this. We use phage display. Phage is a virus that infects bacteria. And there one can engineer them easily. And most importantly, one can easily rescue them from anywhere because they infect bacteria and grow them up. So you make a library of peptides, small pieces of protein, in the coat protein of the phage, which looks like this. It actually looks like a spaceship, so someone put it into the desert, into a Mars landscape there. So you can make a library of peptides that has usually about 1 billion different peptides. The idea is that if you have 1 billion peptides, there are going to be some that have the binding activity you're interested in. And we're interested in, the phage expressing a peptide that makes the phage stick to the vessels of a disease or a vessel that we're otherwise interested in. In this case, I'm showing a tumor vessel. And then by figuring out what peptide the phage carries, we can figure out what peptide has affinity for that vasculature. So this is how it works. You have different shapes, and the one that has the right shape for the receptor in the vessel sits there, and then we can save it from there and use the peptide for our purposes. Now, using this method and drawing from the literature from people who have used other kinds of methods to examine the vasculature, we've shown that the normal vessels in tissues are special in the sense that every tissue puts a signature on its vasculature, even though the vessel lining cells, the endothelial cells, look the same, but they're different. And tumor vessels are very different from normal vessels, and they are an important target of new tumor therapies. Atherosclerotic plaque endothelium is also special. And I'll show you one picture of a peptide that targets Alzheimer's lesions. So diseases put their own signatures on the vasculature. So another twist to this that we discovered a few years ago is that one can find peptides that not only bind to the endothelial lining of the blood vessels, but stick to it and then go through it like this and to a next cell, which may be, for example, a cancer cell, and so on into the tissue. And we call these tumor-penetrating peptides. Here's an actual electron micrograph showing what it looks like. These are little gold particles that are easily detectable in electron microscopy, and they're coated with this kind of a peptide here. And here's the cell surface, and here's a vesicle like that, in the process of going through the cell. So we think that physiologically, the significance of this system is that in normal tissues, if the tissue is in trouble, say a poor blood supply or an injury, it can use this system to bring in nutrients from other cells and from the blood. And this happens quickly. You've all heard about new tumor vessels growing into tumors and preventing this being an important new cancer therapy. Now this takes days, whereas our system, which we call the sender transport system, is instantaneous. So it can provide quick help to a tissue in trouble. And as always happens, if there is a physiological mechanism that can help a tumor, the tumor will use it. So they have hijacked, apparently, this mechanism to help them survive when they grow beyond their blood vessel supply, which they do all the time. So I'd then like to show you a couple of things we have accomplished with this technology. So we use quite a bit we use all kinds of different nanoparticles, but here's one of them that Mike Saylor, who designed this at UCSD, calls nanoworms. And we published the first paper on these nanoworms together, and the reporters all over the world got really interested in this word nanoworm. They gave me a call in the middle of the night and wanted to know what nanoworms were. So it just tells you that it's really important to name something in a way that people remember. So anyway, they are iron oxide crystals in a row coated into a carbohydrate coating that keeps them in place. And then you can attach things to the coating. And we attach usually a linker and then a homing peptide that takes the nanoparticles where we want them, in this case in tumors, and then a drug. And we use a peptide drug also, but it could be a regular drug coupled to the particles. So here we treated, in collaboration with the group at the Salk Institute, glioblastomas. These are extremely malignant brain tumors, and they are essentially untreatable in humans and in mice. And that's what this group, which is headed by Indra Verma, found that they really couldn't do anything about the mice dying like this very quickly. With our nanosystem, we actually ended up curing most of them. And in another model they have that is more aggressive, we prolonged the lifespan of the mice with that nanosystem. And then, knowing that we had room for improvement in this system, we added one of these tumor-penetrating peptides to the system. And now what happened was that while initially we targeted the blood vessels, and they are here, the purple dots in the tumor tissue, and you can see the nanoparticles are red, and there's a complete correspondence between the two, meaning that the nanoparticles never got out of the blood vessels. With this tumor-penetrating peptide, they do, and now we make the tumor cells also a target, not just the vasculature, and we further prolong the lifespan of the mice. They were very impressed at the Salk Institute by these results, and if you'd translate that to human time, it would be a pretty nice prolongation of the time lifespan, even though in this case we didn't get any cures. Now, in another system, we designed what I think is a pretty clever modification of nanoparticle targeting. And that is designing the particles so that they are self-amplifying. One particle finds its target, and then it calls in more particles, which call in more particles, and so forth. So the system self-amplifies. And it's based on blood clotting. There's always a little bit of blood clotting in tumors, in tumor vessels, and not in normal tissues. And that's what it targets first, but then it causes more clotting, which brings in more particles, and so on. And we actually got pretty impressive results with this system. Here's we look at not survival, but tumor volume. So here's how the tumor grows. This is a breast cancer in mice. And here's the treatment group. We essentially prevent tumor growth in all of the mice. And we had 1 out of, I think it was 8, that was actually cured. And that, of course, if you can get a cure of a tumor that otherwise is lethal, even in 10%, that's a big thing. So it's a good system. But interestingly, we did not get anywhere with this in trying to commercialize it. The previous system I showed you is being developed by a company. But this system, the companies are so afraid of clotting that they just wouldn't want to hear about this. So the clever idea didn't go anywhere. That's how it is sometimes. So just one vignette here about Alzheimer's disease targeting, which is new to us. We're mostly tumor biologists and peptide chemists and now nanochemists. But we got involved with a DARPA project where they wanted to target Alzheimer's lesions. And this is a tissue section. And all these blue things are nuclei of individual cells, neurons or other supporting cells in the brain. And this is what is called the dentate gyrus, thick layer of cells in the hippocampus. And this is the area where our brain first starts deteriorating. And this is the area that is particularly hit by Alzheimer's. And we did find a number of peptides that home to these lesions. And here's one of them. And in this case, we have stained the blood vessels red, and the peptide is green. And you can see that there's a blood vessel, and then there's a cell that hugs the blood vessel. And that's what is called an astrocyte. And this is called the neurovascular unit. And so we can now get specifically target things, therapies or diagnostics to Alzheimer's lesions. And we actually were able to show that we can, in mice, do this very early in the disease. And I forgot to point out one thing here. We've shown already that even human, the cells that are derived from a human Alzheimer's patient pick up this peptide. So it works from mouse to human. So that's just a quick summary of what we do and where we hope to be going with this. And the tumor work was mostly done by, and the work on the penetrating peptides by these two individuals and Hongbo Peng, and the Alzheimer's work by Aman Man and Pablo Skodaler. And here's my financial disclosure, if anyone is interested in. Thank you. So I don't suppose there are any questions, and I used up my time anyway.